Welcome to a new episode of Smart Locus. In this episode, we will see regulation of gene expression and lac operon. What do you mean by regulation of gene expression? How a gene expression, or what do you mean by gene expression? Gene expression is nothing but synthesis of protein, right? According to central dogma, the genetic information present in DNA will be transcribed in the form of mRNA, will be finally translated into protein. So a gene will be expressed in the form of a polypeptide or protein. So gene expression, in simple terms, gene expression is nothing but protein synthesis. Okay, so how this gene expression or protein synthesis is regulated? In eukaryotes, there are 4 different levels in which protein synthesis can be regulated. The 4 different levels are, first one is transcriptional level. That means if that particular protein is not required, then transcription of that gene can be blocked. That is, synthesis of mRNA from DNA can be blocked if the protein is not required. Then, processing level. mRNA processing is there now. In eukaryotes, the product of transcription is nothing but hnRNA, heterogeneous nuclear RNA, which will have both exon and intron, right? It has to be processed, which involves 3 steps, right? Splicing, tailing, right? Capping. So capping, tailing, splicing. So at this level, we can regulate the gene expression or protein synthesis. Okay. Then, during the transport of mRNA, see, in eukaryotes, this transcription takes place in nucleus, nucleoplasm, and it has to be transported from nucleoplasm to cytoplasm, where the ribosomes are located for translation or protein synthesis. So this can be blocked if the protein is not required, if that particular protein or enzyme is not required. Then at this level also, the gene expression can be, the protein synthesis can be regulated. Similarly, at last, translational level. Translation of that gene, that information can be blocked or regulated. So, in eukaryotes, the 4 different levels are there to regulate gene expression. But prokaryotes, in prokaryotes, that is in bacteria, Manera, transcriptional initiation is the predominant site for regulation of gene expression. Gene expression. Gene expression is actually, it's meant for the production of a particular protein or enzyme, that is to perform a particular function, right? Enzyme is required to catalyze a particular metabolic conversion. Enzymes are biological catalysts, right? And for example, in E. coli, Escherichia coli, a bacteria, here the enzyme named beta-galactosidase. What is the function of this enzyme? Beta-galactosidase catalyzes hydrolysis of a sugar named lactose. Lactose is a disaccharide. Beta-galactosidase hydrolyzes lactose into 2 monosaccharides, glucose and galactose. That is the function of beta-galactosidase. After hydrolysis, bacteria, E. coli bacteria, will use it as a source of energy. If lactose is not present in the medium, lactose is not there, then production of this enzyme is not required, right? Beta-galactosidase activity is not required. So in such a situation, there is no meaning in producing this enzyme. Gene expression can be regulated here. So we can conclude that it is the metabolic, physiological and environmental conditions that regulate gene expression or protein synthesis. Transcription unit consists of the promoter, then structural gene, then terminator. That is present in DNA, right? So in transcription unit, the activity of the enzyme RNA polymerase, that is the enzyme responsible for polymerization of RNA, that is transcription. So in transcription unit, the activity of RNA polymerase at promoter. Promoter is the site, is the sequence where RNA polymerase binds to initiate transcription, right? Yeah. So the activity of RNA polymerase at promoter sequence is in turn regulated by an interaction between a group of accessory proteins known as regulatory proteins. These regulatory proteins can regulate the process either positively or negatively. If it regulates the process by producing activator, then we call it as positive regulation. Instead, if it regulates the process by producing repressor protein, then we call it as negative regulation. Repressor protein is also known as inhibitor protein. Then what is operator? What is the specific function of operator? So each operon has an operator. Every operon has an operator. So what is the specific role or specific function of this operator? It actually regulates the accessibility of promoter region. Operon concept. This was actually first discovered by Jacob and Monod. They were awarded Nobel Prize for this work also. And they conducted the research work in a bacteria named E. coli, Escherichia coli. And what is operon? Operon is actually a polycistronic structural gene. In prokaryote, structural gene has many cistrons, right? So polycistronic structural gene regulated by common promoter and regulatory genes. Such an arrangement is known as operon. So that is an operon. Okay, so operon is very common in bacteria. Then examples for various operons. TRP operon stands for tryptophan amino acid. ARA operon stands for sugar named arabinose. His operon stands for amino acid named histidine. Val operon stands for valine, another amino acid. Lac operon stands for lactose. Lac operon consists of one regulatory gene and three structural genes. This point is a previous year question, NEET. Lac operon consists of, that was the question. So the right answer is one regulatory gene and three structural genes. That regulatory gene is I. Regulatory gene can regulate the process either by producing activator or by producing repressor. Here this I stands for, repressor is also known as inhibitor. I stands for inhibitor. Okay. So see, this is at the genetic level, DNA level, mRNA level, then protein level. So DNA transcription, mRNA translation, protein. Okay. So here this is regulatory gene. It will code for a particular mRNA which will finally produce a repressor protein. That is the product, repressor protein. Then P stands for promoter. This P also stands for promoter. Then operator. These three are the structural genes. The three structural genes are ZYA. Z gene codes for the enzyme beta-galactosidase, which catalyzes hydrolysis of lactose into glucose and galactose. Then Y gene codes for the production of which enzyme? Permease enzyme, which increases the permeability to lactose. That means this enzyme is required for absorption of lactose into the cytoplasm of bacteria. Okay. Then A gene codes for the enzyme transacetylase. So these three enzymes, the three enzymes are beta-galactosidase, permease and transacetylase. These three enzymes are required for lactose metabolism. Okay. So regulatory gene and three structural genes. Is it clear to you? Regulatory gene and three structural genes. Then this is operator, which controls the accessibility of promoter region to RNA polymerase. This is the promoter region. And here one more P is labeled in NCERT, right? This is the promoter for the RNA polymerase that transcribes the regulator gene. This is the promoter for the RNA polymerase that transcribes the three structural genes. And this promoter is for binding the RNA polymerase that will transcribe this gene. This gene means the regulatory gene, I gene. If lactose is absent in the medium, then the enzymes are not required for the hydrolysis of lactose. The repressor protein will bind on operator. This binding will prevent RNA polymerase from transcribing the 3 structural genes. RNA polymerase enzyme will not be able to transcribe ZYA genes. If there is no transcription, means no mRNA. Without producing mRNA, translation will not occur. As a result, no enzymes are produced. In the presence of lactose, lactose will interact with the repressor, which will make repressor an inactive one. It will produce an inactive repressor. As a result, RNA polymerase enzyme will be able to bind on promoter and the transcription takes place. Transcription of the 3 structural genes takes place. It will be transcribed into a single mRNA. Remember, a single mRNA, which will then get translated into the 3 different polypeptides: beta-galactosidase, permease, and transacetylase. So this is how lac operon works in the presence of lactose. Yes, in the presence of lactose, all the 3 enzymes are required for the metabolism, for the hydrolysis of lactose. So here actually, it is the presence of lactose which induces the production of enzyme. Right? The presence of lactose induces the production of the 3 enzymes. So lactose is known as inducer. So in lac operon, what is the role of lactose? In lac operon, lactose acts as inducer, which induces the production of 3 enzymes. And in bacteria, a low level of lac operon is actually always switched on. Why? Because otherwise this enzyme will not be there in the cytoplasm. Permease enzyme. Without the help of permease, lactose cannot be absorbed into the cytoplasm of bacterial cell. So we can conclude that a low level of lac operon is always switched on in bacteria. And the regulation discussed here, regulation of lac operon discussed here, is an example for positive regulation or negative regulation. This is actually negative regulation. Why? Because the regulator regulates the whole process by producing repressor. If it regulates the process by producing repressor, it is known as negative regulation. Instead, if it produces activator, then we can call it as positive regulation. If you have not yet subscribed the channel, please subscribe and press the bell button also. Thanks for watching.